All right, now we're going to be talking about the brief section on cartilage growth in the skeletal system. And the primary objective here is to compare this interstitial and appositional growth of the cartilage. So the process of cartilage growth, it begins during embryologic development, and we have interstitial growth, which is growth in length. It occurs within the internal regions of the cartilage, and the appositional growth, which is the width, and it occurs on the cartilage's outside edges, outside edge. Zoom in here. So we have here on our picture here looking at the interstitial growth specifically and occurring within hyaline cartilage, and we have here our matrix, and then we have our chondrocyte within the lacuna. Look at those specifically. So with interstitial growth, the chondrocyte within the lacuna begins to exhibit mitotic activity, ready to split, right, and divide. And we have lacuna on the outside here, that outer layer, and then the chondrocyte itself with the matrix, the matrix outside here, the lacuna, and then this being the chondrocyte nucleus there. And two cells, now called chondroblasts, are produced by mitosis from one chondrocyte and occupy one lacuna. Then each cell produces a new matrix. It begins to separate from its neighbor. Each cell is now a chondrocyte, and cartilage continues to grow internally as they begin to separate into separate chondrocytes. And appositional growth, more in width. Now we're looking at the perichondrium here. And mitotic activity occurs in the stem cells within that perichondrium, right? We have the perichondrium layer here with the mesenchymal cells specifically, and these dividing undifferentiated stem cell example here. And then new undifferentiated stem cells are committed to cells that differentiate into chondroblasts, and then they're formed. These chondroblasts produce the new matrix at the periphery. So you have these undifferentiated stem cells here, and the committed cells that are differentiating into chondroblasts. And as a result of that matrix formation, the chondroblasts then push apart and become chondrocytes. And these chondrocytes continue to produce more matrix at that periphery. And here you can see the undifferentiated stem cells here, with the chondrocyte seeking its new matrix now, separate, or secreting new matrix, excuse me. So, where do interstitial and appositional growth of cartilage occur? That's a simple question you should be able to answer. Bone formation. We're going to identify bones that are produced by intramembranous ossification. Explain the 4 steps. You should be able to explain the main steps, 4 main steps in intramembranous ossification. Explain the steps in endochondral ossification of a long bone, and differentiate between intramembranous ossification and endochondral ossification. So with ossification, which is osteogenesis, the formation and development of bone connective tissue, it begins in the embryo. It continues through childhood and adolescence. By 8th through 12th weeks of embryonic development, the skeleton begins forming, forms an intramembranous ossification or endochondral ossification. Intramembranous ossification is bone growth within the membrane. It's also called dermal ossification. It produces the flat bones of the skull, some of the facial bones, the mandible for the jaw, and the central part of the clavicle. For intramembranous ossification, ossification centers are formed within the thickened region of the mesenchyme. Some cells become osteoprogenitor cells, like we talked about before. Some cells become osteoblasts secreting osteoid. Now, osteoid undergoes calcification, with calcium salts depositing onto the osteoid and crystallizing, and then entrapped cells become the osteocytes. And then woven bone and surrounding periosteum form. At first, bone is immature and poorly organized. Woven bone is primary bone, and the mesenchyme surrounding woven bone forms the periosteum. And lamellar bone, secondary bone, replaces that woven bone, and the compact spongy bone then forms from the trabeculae. And this is typical structure of a flat cranial type bone as an example, composed of 2 external layers of the compact bone with a layer of spongy bone in between. So here we can see, looking at our flat bone of our skull here, the ossification centers forming within that thickened regions of that mesenchyme here. And we have this being the ossification center itself, the collagen fibers, the osteoblasts, mesenchymal cells. The osteoid undergoes calcification, okay? And with the osteocytes here, the central area, and the osteoids, the osteoblasts, and this new calcified bone matrix. Woven bone and then surrounding periosteum then form. Here we can see the blood vessel, basically bringing blood to that cell, to the cells. And the trabecula of the woven bone. Mesenchyme condenses to form the periosteum outside here. And the lamellar bone replaces the woven bone as compact and spongy bone form. So we can see that compact bone now forming, and the spongy bone as well. Lamellar bone out here, and that periosteum on the outside. Endochondral ossification begins with the hyaline cartilage model. It produces most of your bones of your skeleton, including your upper and lower limbs, your pelvis, your vertebrae, and your clavicle. An example of this process is long bone development. And the steps of long bone development in a limb, the fetal hyaline cartilage model develops with the chondroblast secreting cartilage matrix, and 8th to 12th week of the development is when you start to see that significant progress. The steps of long bone continuing here with the cartilage calcifying, the periosteal bone collar forms. Chondrocytes in the cartilage model produce holes in the matrix. The matrix calcifies, and chondrocytes die. This produces the calcified cartilage shaft with large holes. Blood vessels are growing toward the cartilage, and the osteoblasts develop and secrete osteoid. That forms a layer of osteoid around the calcified cartilage shaft with the periosteal bone collar formed. The primary ossification center forms in the diaphysis, and the periosteal bud extends from the periosteum into the cartilage shaft. The growth of capillaries and the osteoblasts occur here. Osteoids produce osteoid on calcified cartilage template. That's the primary ossification center. It's the first major center of bone formation, and most is formed by the 12th week development. Bone development extends in both directions toward epiphyses, on both ends of the long bone in this example, and the bone connective tissue displaces the calcified degenerating cartilage. The secondary ossification center form in the epiphyses at the ends. Hyaline cartilage calcifies and degenerates, blood vessels and osteoprogenitor cells enter, and then secondary ossification centers form as bone displaces that cartilage, and not all of it's going to be formed at birth. Osteoclast resorbs some bone matrix, concept we're going to see over and over again with talking about the skeletal system, and it creates a hollow medullary cavity. Bone replaces the cartilage, except the articular cartilage and the epiphyseal plates. The epiphyseal plates ossify, and they form these epiphyseal lines, and then lengthwise bone growth continues into puberty. Growth continues until epiphyseal plate is converted to epiphyseal line, which occurs in sexual maturity, and indicates the bones have reached adult length. Technically, if you break that epiphyseal plate, if that some damage occurs at that epiphyseal plate, it could affect the growth of that particular bone. It's rare, it's unusual, there's enough protection around the area, it's highly unlikely to be the case if a bone break occurs in a child, but technically if you damage the epiphyseal plate, that bone would potentially be harmed in its growth. You could have a malformed bone there on that one side, because you're assuming you have same bone on the opposing side. If we're talking about like the femur or the humerus, which are the most common bones where you see damage of this type. But that said, that reaches adult length, that's somewhere around sexual maturity, and then in which case that epiphyseal plate converts to that epiphyseal line. And it occurs between ages 10 to 25. It's unusual to see it happen as early as 10, typically more like 13, somewhere in there for some girls. Could be earlier, but typically somewhere in there, 13 to 17, somewhere in there. Whereas the men can be later and typically are, but can vary between individuals, of course. So at the epichondral ossification, so the 8 to 12 weeks, with the perichondrium here with hyaline cartilage at the end here, the fetal hyaline cartilage model starts to develop at this point. The 10-week fetus, you can see in this example here, special staining highlights that cartilaginous models of the bones, and arrow points to specifically the humerus. There's the humerus right there. So in that fetal period, there's a deteriorating cartilage matrix, that periosteal bone collar, the hyaline cartilage. The cartilage calcifies and periosteal bone collar forms around that diaphysis. And the epiphyseal blood vessels are seen, of course, at the ends here. And then the periosteum forming here, the blood vessels, the periosteal fluid entering in here. Primary ossification center forms in this diaphysis. Then the secondary ossification centers form in the epiphyses. The epiphyseal blood vessels at the end here, developing compact bone in the medullary cavity. Along with the calcified cartilage now. Here you can see a skeleton of a neonate. The child, so we have the articular cartilage, of course, on the outer portion at the proximal and distal ends, and they have the spongy bone here, epiphyseal plate, again the growth plate The periosteum, the outer layers here, and the diaphysis, the medullary cavity, the central cavity, and the compact bone surrounding that central cavity. And that bone now is replacing the cartilage, except the articular cartilage and the epiphyseal plates. That's not being replaced. The articular cartilage, I'm sorry, the epiphyseal plates here are ossifying and forming the epiphyseal plate more completely in the later teens toward adulthood. Obviously, that bone length you'd expect to be growing significantly in length for sure, right? Until those epiphyseal plates become epiphyseal lines. And the great example of that is, of course, the humerus. They have the unfused epiphyses in the diaphysis, where you're going to see those fuse as they grow into adulthood. So you should be able to answer then, when does the intramembranous ossification then begin? What bones are formed from this method? And then briefly describe the process by which a long bone forms by endochondral ossification. And we'll talk about section 7.5 here shortly.